Good afternoon, everyone. Welcome to the H3D Foundation webinar series. Our today's speaker is Dr. Wynand Symthe. Dr. Symthe is Associate Director at Pharmacometrics Division of Certara, a drug discovery and development consultancy providing various services, including pharmacometrics, software, including Phoenix, pharmacokinetics, and pharmacodynamics platform, and resources, including professional certified training. Dr. Symthe is a clinical pharmacist and pharmacometrician. He is interested in the PK/PD relationships of antimicrobial with the special focus on TB and malaria. He has over a decade experience of clinical trial and has helped develop and chair the antimicrobial stewardship AMS program at a private tertiary hospital. Before we start with the talk, the general housekeeping rules. We'll have a Q&A session at the end of the talk, so questions can be posted in the chat or kept for the end of the talk. Today's topic is Certara, working together to advance drug discovery in Africa. And now I would like to request Dr. Symthe to share his work with us. Over to you, Dr. Symthe.
Good afternoon, Shruti, and good afternoon to the viewers. Thank you for the very kind introduction. And yes, very, very happy to be here, and a big privilege to be able to talk to an audience specific focus within Africa. So, Shruti, I just want to confirm before we start, the audience will be able to see my screen at the moment?
Yes,
okay, so
everybody
presentation
can. Yeah.
Okay, perfect.
Yes.
Okay, so really off the bat and talking to why we're doing this presentation is essentially really to connect drug discovery programs within Africa. So now Certara, as introduced, a drug consultancy company, which basically includes the span of drug discovery. And here we can look at, for instance, various aspects of drug discovery, from early development or discovery all the way through to submission processes. With this, I must say that I'm employed within the company, within their African branch. And I suppose we go right here from the start. Let's just have a look if my screen share is moving. Let me get this. All right, so have a look there. There we go. Okay, so from the map over here, we can see it's obviously like most companies, unfortunately, it's very northern hemisphere dominated, but there is a concerted effort to actually expand this program within African countries. And from the map, you can see that now in Africa, there's at least two branches, that being there of Cairo and Cape Town. And essentially, we've now got under directorship, senior directorship from Samir Muksasi, we've got 4 people, so I think 2 in Cairo and very newly introduced now 2 in Cape Town, with a long-term process of expanding this program within other African countries. So as a consultancy, as I mentioned, it's not just the early development here with pharmacometricians in the company. This spans a range of regulatory experts, economists, software and technology. So it really spans the whole process to try and help move a drug from early development right the way until you actually submit the drug on the market. So as you know, the drug discovery pipeline, everyone talks about time is money, and you know that's no different here. And essentially what the company does is really kind of integrates the personnel across various spheres to try and basically work smarter and faster. All right. So we typically knowledgeable of our clinical pharmacology side, the pharmacometricians, you know, and physiology side, but we integrate with various other softwares and technical skills experts. You know, from the regulatory writers all the way to submission, and we really try and optimize this process from discovery all the way through to submissions. And then just to kind of delve a little bit deeper into that, we have various aspects of support, as was mentioned earlier, and not only in the sense of, if we look across this pipeline from discovery on the left-hand side here, It ranges all the way through to the post-approval or post-marketing type studies. And then we also stratify here across software and technical services rendered by the company. And one thing I just wanted to highlight, maybe to show, is that a software and technical service called Symthe really spans across this drug discovery pipeline. So you can see it's involved in early aspects of discovery all the way through to post-approval processes. So that's just something to keep in the back of the mind as we deal with that later in the talk. This I just wanted to highlight. Now I know that the slide says why H3D should partner with Certara, but that's no different to any other local-based companies within Africa. And it's really that we share this common goal. I think that's really what I want to stress here. Certara really is about obviously being consultant, providing expertise support, but really at heart, what we're trying to do as well is that we're really trying to not only train or develop drug discovery scientists, and Certara specifically pharmacometrician, but anywhere in the drug discovery process, but also provide employment. It's one thing to train up a student and get them expertise, but then all too often we see this employment, seeked overseas. So essentially, this what we call a brain drain. And really, we don't have to have that, and we should take initiative and see where we can start employing these scientists locally, and really keep this critical mass in the countries that where we can actually develop drugs and market them ourselves. So, as I said, we're going to delve into a couple of these aspects of drug discovery and softwares. The one that I wanted to start off with is more the modeling and simulation process, where we actually use it more to optimize trial design. And I'll go a little bit into that, because typically we normally see pharmacometricians involved in more just population models, PK, PD, that sort of thing. But yeah, we can see often we start off with a research question in mind. Okay, so typically clinically based, but it really can be in any question base. We then obviously have a population of interest that we want to study that in. And then we have a particular trial design that we're thinking of how to actually study that. We put that together, and then we ultimately end in with a clinical trial design. So how are we actually going to study this within our population with a particular question in mind? We run the study, and then from that, we get this data output, all right? So once we have all the data, we collected the data, we then put it into a model. And a model is really just a mathematical explanator of that data. Okay, so why do we want to do that? So having this mathematical knowledge that incorporates all this trial design, we're then able to ask these what-if questions, okay? So in this particular design set, for instance, we used a particular dose, or we used a particular population, or whatever the scenario was. The model then allows us to ask these what-if questions, like if we had to have a different dose, if we had to have a different design, or a different population, or some sort of covariate that was changed. One part that I do want to move focus onto is the design part, the trial design. So we focus in on here, we can use the model and various simulation modeling practices to look a little bit further into sample size. Now, this is obviously a question that comes up time and time again. Do you have the optimal, sample size for your study? So this is one of the tools we can actually use modeling for. And power obviously depends on, your number of subjects required. The effect size. So if you're looking for a relatively small effect size between, say, two different drugs, you're going to need a large sample. The study design itself, so optimal designs can kind of minimize that number. So think of like crossover design studies versus your typical parallel studies, possibly more numbers needed there. And then also the type of error that you're comfortable at applying. So obviously, if your type 1 error, if you want type 1 error, then obviously your sample size needs to increase. Now that sample size is not just your typical n equals whatever number. in other words, subjects, but it can also be the number of arms in the study you imply, or the number of sampling, occasions that you have per patient. So it can get really complex. So what we do then is to actually employ the help of these PK models. So this model can be, as I mentioned, PK based, but it's PK, PD, whatever type of model you have. What we do is then we simulate this data or this design that we had, and we run this multiple times. Okay, so we say, for instance, we do looking at sample size now. So we run it, and under the aspect of we include a certain sample size number, and then we do the test. And we replicate this many times to see the numbers and see the kind of variance that we see within that. So once we do that, we can then repeat this process using a number of different designs. And in the simplest case, maybe different sample numbers, sample size. Once we do that, and we run the test multiple times under multiple different scenarios, we can actually start optimizing on this particular outcome of interest, this test that we want. So we can really design it specifically to look at those scenarios. What we do in the end is obviously we summarize all these scenarios and replicates, and then we can come up with suggestions. So as I said, in the simplest aspect of it, we can look at the optimal sample size required to detect a certain difference in a study. However, this can be applied across different scenarios, and you can actually look at different trial designs for different outcomes. And often what happens is that when you're doing these replicates in different scenarios, we come up with new ideas. You think of new designs and think of new concepts. So it's a very useful tool. And I know my time is pressed, so if there is a bit of time near the end, I can maybe squeeze in an outcome or practical example of these simulation-based studies where it's applied in, say, pediatric populations. But as I said, we can always come back to this if there's time. What I then wanted to touch on was also an overview of Symthe. I said that earlier in our talk. And the reason being is that this software and technical support actually spans the range of our drug discovery program, right? So it goes right from early drug discovery development all the way through to submission phase. And specifically in Certara, where these expertise and skill sets are applied, it's really integrated optimally across the different consultants in the company. So we typically think of these as our PK/PD or quantitative scientists. If we look in a little bit in more detail, most people would probably think of PBPK as or Symthe as PBPK, but actually there's a whole lot more that you can flesh out on that. And as you can see on the schematic here, it's not only the PBPK aspects across, but there's in pediatrics or animals or different design aspects, but it also includes your quantitative systems. So the QSP, I'm not sure if some of the audience have heard of this, but essentially this is different PD markers or PD models that you can apply to. And one of the more recent advances or applications of Symthe is looking into what we call this safety and toxicity aspect. So it's referred to as the QSTS, Quantitative Systems and Toxicology Safety Designs. So not only are we interested in the physiological or mechanistic parts of the models or the quantitative parts of it, which is more the what we call the on-target or specific targeted aspect, but also the side effect profiles. In other words, the off-target hits that our compounds have. And this software is very useful to actually look into aspects of those. Okay, so it's a very more mechanistic modeling process in comparison, say, to our population type models, where it's very typically non-mem style, so simplified type of compartmental modeling. If we then have to look into how these PBPK or Symthe are applied, and it's applied very similarly in the biosimulation part, is that you can then start looking specifically into systems. So essentially combining all sorts of tissues and parts of the body where you want to try and optimize the drug and drug exposure, but also at certain endpoints. In other words, if you're looking at, say, first in human type dosing, dose escalation type studies. But more specifically, I suppose in early phase, you can look at drug-drug interactions, maybe even I suppose what's applied locally is also bioequivalence type studies can also be very, very useful in that. And then very typically early on in your drug discovery, you want to also look at certain food effects. So compartmentalizing this system or making this in silico type human really enables you to use all sorts of data. And maybe the next slide can help me out a bit here. I'm not sure if this is too clear, but maybe if you look at the left-hand panel, you can basically see where typically in, as I said, mentioned in population type modeling, we really kind of simplify the system into, say, certain compartments, 1 or 2 or perhaps 3 compartments. But in PBPK type modeling, we really try and mechanistically explain the physiology. So here we can see typically we either dose into the gut and then feed into the various tissues or organs, or else we can do an IV route. We push that straight into the venous supply, and then this gets circulated into the various tissues. So we can apply all the data that we can extract, say, from these in vitro, in vivo systems, and then apply these endpoints that we measure within our models, within these PK types models. And then we can quantitatively study the concentrations that we're going to be exposed in these various tissues. Okay, and that can help us out explore various safety aspects, but also on-target aspects as well. So it's a very nice modeling structure, modeling system to really explore the compartments of the modeling, and we can feed in a lot of data to that. Then just looking at, again, highlighting before I move on again, this SymSip system really is integrated across the drug development system. So from your early discovery, where we're looking at, say, your preclinical, more your animal type models, your laboratory type tissue organs, we can apply all of that data, start building our mechanistic model from all of that data. Then working with this model, we can transition through into our early development phases. So your early phase studies, first in human studies, typically looking at dose effects, typically looking at some sort of food effects studies early on, all the way through to our, what we typically call our post-marketing studies, where we have a registered drug, perhaps or more often in say adult populations, typical adult populations, should I say. And then we want to start looking at how we can actually use this drug in study populations that we haven't looked at. So in other words, pediatric type populations or other vulnerable populations, be it pregnant women or so forth. And we can use this data as we collected it and then apply our knowledge on what these other covariates are, and then look at this, how this model can predict into those populations. So a very, very useful tool in drug development. One aspect that I suppose is not really touched, well, it depends who you ask, I suppose, but certainly for me, an aspect of pharmacometrics, or at least population type modeling, we all learn to use a particular tool or software, and we all too often want to jump in and model straight away. But we really have to question and look at the data and really see what the data is telling us, okay? And in this case, EDA, or exploratory data analysis, is really looking at the data and seeing exactly, the distributions, the patterns, what is there, and what questions can we actually be asking and using the data for. Okay, so we often design the studies, collect the information, have these data sets, but then we need to explore this data set even before we start applying modeling techniques. Okay, so it's a real, and hopefully for the next few slides, I can convince you that this is important. So if we look at this, now I'll allow you some time to be mesmerized by these moving dots or plots. But essentially, if you have a look, we've essentially got data on our y-axis, okay, and data on our x-axis. And if you look at in the gray box on the right-hand side, you can see summary statistics. Here we give the mean of the x, the mean of the y, we give a variance or standard deviation, a spread of the x and the spread of the y, as well as a correlation. Now, all of these plots that you see all share the same mean, the same standard deviation, and the same correlation, but yet produce these very different distributions. Okay, so if we think about it as how to apply or what we're going to do with the data, it doesn't help just looking at summary stats, say, for instance, of the data. We actually got to look at visually the representation of the data. Okay, so it's really important to actually visualize our data. So one of the tools that would be useful, that I found very useful, and certainly this is a software that was developed by our head director in Pharmacometrics Africa, that's Certara Muksasi. And here you can see the link to on the GitHub repository where you can access this software. It's freely available, okay, through an R-Shiny application. Those of you not familiar with R-Shiny, I suppose I can explain. It's really just an interactive software. So essentially, you load the software in R, and you get this user interface that you're then able to use. So maybe with the next slide, I can show you. Oh, here we go. Okay, so this is the user interface, or UI. Yeah, we can see that you've got this plot highlighted, essentially with the, I think it's what we've got, RIF on your y-axis, time on your x-axis, and there's some distribution of concentration time profile across the time profile. But we can vary this, and you can either use the libraries that you can actually load for training purposes, or you can load your own data and play around with that. The software does provide vignettes, or what we call, I suppose, instructional video type trainings. So you can really explore possibilities of what you can do on this software. And then just as a brief summary, I mean, you can look at stratifying your data. In this case, I think it's a formulation that they're spreading it on. And immediately you can start picking up on, maybe there's particular outliers that you're looking at. You start seeing a whole lot of things once you start playing around with your data. And that's essentially it. You've got to explore the data. And you can do it across various designs. I mean, you can color stratify. You can, essentially plot individual profiles, so you can see individuals that maybe stick out, whether they stick out at formulation issue or whether they stick out at concentration issues, wherever it might be. You can really, you can manipulate this software a lot, and it's just the user experience. I think the limitation is more on the user part. So it's really questions that you want to ask. And then just to visualize that, I mean, this is an example of really how much you can actually look at the data. So it's not typically just your continuous type data. You can look at all sorts of, covariates that you typically find in your data set, be it categorical, like your gender and race or formulations. And then you can look at that in relation to typical things that we see in the data, like age, height, weight, and so forth. And really visualize everything that your data is telling you. And this really helps optimally design your research question that you have in mind. And you can already see what you can be expecting. So when you start getting results from your models or from your questions, it's going to start making sense, or it should mirror what you're seeing on the plots. Okay, so that's really, hopefully I've now convinced you that this is a very, very useful tool. So going from one very useful tool to another, again, the author of this one is our senior director in Africa, Certara Muksasi, and he developed a covariate thing called CovFX plot. And really, this is a software that allows effective communication of covariate findings. So I'm not sure how familiar you are with certain publications, and often where we see covariate effects summarized as, the food effect on clearance was 0.5. What does that mean? Okay, so just by example, I'll give you I think this is from a Duranover example. So it's on table output. And if I have to ask you, how much time do you need to read through and understand this table? Okay, I think a substantial time is probably needed. And if I have to ask you a further question, is like, which concomitant drug increases exposure the most? And which one decreases exposure the most? And are the effects similar on all exposures? How much time would you actually need to answer this question? So actually, it's going to take a while, right? It's possible, but you're going to take a while. Whereas if I have to counter that with the COVFX output plot of the same data, you can immediately start seeing. Now, hang on, look, if I have to start looking at this data, where we can look at types of drug classes. So yeah, we can see in the top panel the protease inhibitors, whereas other antiretrovirals on the bottom part. Clearly, the protease inhibitors. Here we see the plots of exposure. So in this case, it's AUC with squares. The Cmax as a exposure in diamond, and the semen or trough, which is obviously the more common endpoint exposure in antiretrovirals that we look at. Those are in the circles. So if we look at the plot here, with 1 being our typical value, okay, the typical value in the data set, in relation to the covariate effect. In other words, the covariate effect, we're having a look at concomitant dosing with certain drug regimens and doses. How does that change our exposure? And now, in using the forest plots design, where the median effect is shown on the blue dot with its 95% confidence interval using the line. So shifting, so let's have a look at the main culprits. Main culprits, lopinavir, ritonavir. Okay, now obviously those involved with a bit of Duranavir would understand or have not prior knowledge of this, but essentially, yes, co-administration with lopinavir or ritonavir, this is going to influence the exposure that you're going to see with the Duranavir. There may be some Duranavir exposures changing a bit, but certainly the main contributors sit over here with the protease inhibitors. And then what we do in addition to that, although I will explain this a little bit further, is put a bit of a clinical significance marker on it. So those typically involved in bioequivalence studies would understand if we are looking at a relationship of two exposure variables, we're looking at a ratio. And if that ratio falls on 1, it means it's exactly the same. However, if you have a ratio greater than 1, up to 1.25, that's obviously having greater exposure in the test. Or being less than 1, it means you're having exposure less than the typical value. Okay, so these typically decrease exposure values. So then, not only is this a reasonable communication of it, so much so that even industry now has guidelines using with recommendations saying forest plots should be used to illustrate this effect. Okay, so if the regulators are starting to do this, you should know this is the important part. Okay, so now when you're trying to submit your models and data to try and register a drug, we're going to actually need these sort of outputs, okay, to communicate effectively. So that's another reason why we should actually be using and learning these tools. And walking you through this example, we just got a simple forest plot looking at the effect of weight and sex on AUC. On the left-hand side, we can see the weight in the upper panel, sex in the lower panel, in relation to their typical value in the data set. In this case, this would be a female in relation to a male, and this would be, the median weight in relation to the quartiles of weight. So in this example, obviously a standard dose of, say, Duranavir, you give that to a low weight patient, you're going to get a high exposure. You give that same dose to a high weight, and you're going to get a low exposure, right? So it's just very easily seen that weight is a significant covariate that you should account for in the Duranavir example. Okay, so that's in simply what the COVFX plot can do for you. But not only visually, it will also have obviously a table output. You can actually see the same data in table. In addition to that, it also has an R output. Okay, so I mentioned this is an R shiny output, puts it all into an R code, of which you can then replicate at a later stage. Okay, so it's something that you can keep a local repository of. If we then had to look at, well, what influences this spread from the typical value? Okay, so this is an important aspect to understand. So if we're looking in this example, we're looking at the parameter clearance. Okay, so in this case, it happens to be 4, and that would be obviously in some sort of volume per unit time. If we include a standard deviation, so this would obviously come with some sort of variability, we can see the standard deviation is about 0.4. So the typical value here would be 4 plus minus 0.4. Okay, so that would be our distribution. That's just using the uncertainty in the actual parameter estimate. If we then had to include a covariate effect on that parameter, like for instance weight, and this is typically a scaling parameter that we use, we can then see in this case we look at clearance with weight standardized to the typical weight in the data set. This happens to be 85 kilo, and then we do allometric scaling on top of that. In other words, power to the 0.75. What is effectively we've done is we took the spread that you see from a typical value of clearance, and we've widened that a little bit further. Okay, so now we're not only taking the variance of the spread of the estimate of clearance, but also now the effect of weight on that parameter. And then in addition to that spread or uncertainty, we can also see that uncertainty introduced with the added between subject variabilities in our data set. So I'm not sure who in the audience is more non-mem orientated, so that's me. Then we talk about ETAs. Okay, so ETAs is just really the between subject variability, or IIV, the intersubject variability. And there we can see that also further spreads the uncertainty in that parameter estimate. Okay, so that's what can make up our forest plot, that spread bar that we see. So to visually see that, we just look at the lower left-hand panel now. We can see a clearance value of 4 typically included there. And then the equation tells you there clearance is described by the typical population clearance. And that's scaled to the weight, and the average value in the data set being 85. As I said, the power comes into that, so that's just the allometry applied to it. And then in addition, the ETA is included. Okay, so that introduces the extra spread. So yeah, we see in the purple, you've got this more narrow spread of distribution. Adding the covariate effect, in this case happens to be weight, you increase the spread a little bit, and then including the BSV, you see even more spread. Okay, so these are just to illustrate the spread of the data that we see in these forest plots. And then to make it more understandable, to make it more comparable, we just scale this to a standard value. In this case, happens just to be 1. So now we can visually see this. You know that I think it was a dotted line in our forest plot that we saw. That's your standardized value that we use in the CovFX plot. Okay, so this happens to be represented by 1, and you can see the spread across that. Okay, so now we take this a little bit further, and we see really how it's, and don't worry, you don't have to follow this. It's just a procedure of what really happens in the background with the CovFX plot. And essentially, it's just really looking at the covariates one at a time, and you basically manipulate the one covariate by keeping all other covariates the same. And then you look at the distributions of those. In this case, I think we're looking at concentration time profile one at a time. And I think this is what the typical female across weight ranges as you change it within the class of this patient, I think having an albumin of about 45. So you do this many times over, and you replicate all the covariates out. You then include your variability on those profiles. So those are the shaded areas. And then from those exposures, you then actually capture typical exposure parameters, and those being either a Cmax, a Cmin, or an AUC. These are just easier clinical endpoints which our clinicians can use. And then visually we can show that on our plots. Now either you can use these typical like Gaussian distribution curves, or what we've seen using in our forest plot, the simple dot with a line through being the 90% confidence interval, whichever confidence interval you want to display. And in this case, we happen to be showing the AUC, but as you saw, we can actually look at various clinical endpoints, either show AUC, Cmax, or Cmin, or even show all of them. Okay, so it's just a question of what was the question initially. Okay, So the output that you see, essentially this is it. This is the user interface showing you across all aspects of the CarbFX plot package. And here we can see weight is on the top row into its quartiles of weight, then followed by creatinine clearance, again pushed into various quartiles. The dose, I think there's 3 dose levels here. The sex distribution. So yeah, again, in this case, the male is the typical value in the middle. Female showing how much different it is from the male, and then also the formulation where the capsules are typical and looking at tablet in relation to the capsule. And then the shaded area, as I mentioned, that's just the clinical or relevant clinical endpoint distribution that we typically look at. So yeah, very, very simply, you can see, okay, well, actually the important covariates here to look at or to keep in mind is first of all dose. So you can clearly see the outlier here on relative to the 4 times milligram. The 1 milligram dose really is as far as Cmax is concerned, because that's our parameter, that's our exposure parameter we're looking at. That's really low. And then the creatinine clearance also explains exposure in this case as well. Okay, so that's as easy as it gets. Okay, so it's really easy to communicate these findings to our audience. And I think I'm running a little bit out of time, so I'm going to I think it's another 3 slides or so, and then I'll be finished. One of the most useful tools or exposures that I've had while working with Certara is working with the what they call DAC, or Design, Analyze, Communicate tool. This is in partnership with the Bill and Melinda Gates Foundation. And essentially, this tool is really about trying to improve trial design. Okay, so well, not only trial design, but basically clinical outcomes. So how does this actually work? Now, remember, this is a freely available, I think on the previous slides, I'll go back to that, the Global Health Network. You can see there's the link that you can actually go to on Global Health Networks and get access to this tool. Essentially, what the Bill Melinda Gates Foundation uses this DAC tool is to review protocols that are submitted to them. Okay, so you can think of funders, and typically in Africa, I think we can think of the big funders being possibly EDCTP or WHO or various donors that lend money into research in Africa. They can also then start applying this tool. And one of the useful things of DAC is that DAC is now not only originally designed for Bill and Melinda Gates, but now there's a DAC Africa that's now started, and I believe it's in Senegal, but I stand to be corrected. So, but yes, it's now starting to apply across various platforms across the world. Again, so what is DAC? So it really it's a set of questions that are asked to the PIs, or the people who develop the protocol, and it's to improve this concept of informativeness. Okay, so what is informativeness? Okay, so every study will have a particular outcome. Okay, it might be a positive outcome in which it has some sort of positive result or a negative result. It doesn't matter if it's positive or negative, but it is important if it's informative. In other words, if it actually confirms this is a study and it has an outcome that it can actually have evidence for it. So either it has strong evidence for the drug activity or not, but it's not one of those that end in the typical scenario where the final conclusions are, we needed more patients to power the study, so we really can't say anything. Okay, and this is it. So this particular tool, and I will go a little bit more into this on the next slide, or my last slide, where the DAC assessment tool really looks at aspects of this protocol that's submitted to them under general aspects, design aspects, analyze aspects, and communication aspects. Now, this is usually submitted to a review team. Use this tool to actually review or critique the protocol, and really to give guidance to the protocol developers to say, okay, have you considered these aspects to make the trial more informative? So as I said, it's really a tool that was used by Bill and Melinda Gates for protocols submitted to them, but it can also be used by the individual. So yes, you don't submit it to a reviewer team to use the tool, but you yourself can use the tool. And it will just help you by asking questions, make you think, have I considered these aspects in developing or writing my protocol? So hopefully with time, we can get funding agencies within Africa to apply these tools to the grant funders or the people that fund the studies within Africa. The more we can show that we've run informative studies in this country or in this continent, the more likely we are then to see investment within drug development within this country and within this continent. And that's really the ultimate outcome that we want to see. And with that, I'd like to thank the audience and thank H3D Foundation for inviting me to give this talk. And my acknowledgements obviously go to Certara, the company I work for my senior director, Samir Muksasi, for exceptional guidance and obviously support, and also the Bill and Melinda Gates Foundation for showing off the tool, the DAC tool that they used. And with that, I can accept questions. Thank you.